All right, so today's activity is an actual lab here on evidence of evolution. So I want to take you through a kind of a brief explanation here. So one of the things that you really have to pay attention to with this is the different vocabulary in each section, and make sure that you use your notes as a reference in terms of what exactly is it talking about. So the first thing we're going to be talking about are homologous structures. So that's one of your vocabulary words, and if you don't understand what that means, you need to go back to your notes and remember what that means. So this is going to deal first with these set of structures here on the second page. I'm just going to zoom out a little bit so I can get them all on one screen. All right, so this is the human arm bones, and you can see here we've got labeled the humerus, the radius, the ulna, the carpal, metacarpal, and phalanges. And you'll also notice that the humerus has a particular pattern, the radius has no pattern, the ulna has a pattern, and the carpal bones have a pattern, and then again the metacarpal and phalanges don't have a pattern. So that corresponds to excuse me, the different patterns on the different bones you see in the other organisms. So your first job is you're going to need 5 different colored pencils. So you're going to color the humerus. Let's say you color it pink. Well, then when you come over to the whale, you're going to find the humerus here, and you're going to color that bone pink. And same thing with the crocodile. And then you're going to find the humerus here on the cat and the bird and the bat, and you're going to color all of their humeruses pink. And then you're going to go back and you're going to do the same thing with the radius, the ulna, on all of them. Now, all of the carpal bones, the wrist bones, get one color. And then all of the metacarpal phalange bones, those two sets together, okay, get a single color. So I know some of the carpal bone shading is difficult to see, so if you have questions, please ask. All right, and try to pick colors that I can actually tell the difference between them, okay? Don't leave it up to my judgment when I'm grading it. All right, and so then you've got to describe the function of each set of bones below. So what do we use the human arm for? What does the whale use its arm for? The cat, the bat, the bird, and the crocodile. Then you want to look at those. Are they arranged in a similar way? Okay. These structures are formed in similar ways during embryonic development and share like arrangements. However, they have somewhat different forms and functions. They are called homologous structures. Okay, so there may be some clues to some of those answers there. The second set is analogous structures. Okay, and you actually want to read down here first. Some apparently unrelated organisms or animals have organs with similar functions, yet are very different in structure and form. These structures are called analogous structures. And we're going to be looking at the butterfly wing and the bird wing. Now the bird wing very clearly here has bones in it. We just colored bones in that. Now what you have to understand is butterflies don't have bones. These are not bones. These are simply strips of tissue that you are seeing. So don't get that confused. So you want to talk about the function and how are the structures different, okay? And then talk about any other structural similarities that would suggest that they are related. When it talks about the word taxonomically, that means through evolution. Okay, so do they share any structural similarities that show that they would be related through evolution? The last part of this is about vestigial structures, and we're going to look at the cavefish and the minnow here and how they are related, but the cavefish is blind. So the questions are actually at the top of the next page, kind of looking at this, and that you want to do organs or structures that have lost their function in the organism and have become reduced in size because of efficiency are called vestigial structures. Human vestigial organs are well documented. So these are human vestigial structures. Okay, they become reduced in size and they have lost their function. So try to come up with a probable function. You could probably do some research with this on your computer. And why vestigial? Why did they become not useful to us? Okay. Then you've got some analysis and interpretation questions. Okay, one on homologous structures from part one from analogous structures from part 2. And then you have to list 2 structures that are not on table 1 that you think are vestigial, and you have to explain why. Okay, so hopefully this is a pretty simple lab, although it is one that makes you think, and you should not rush through it because you will not do well if you rush.